This conference will now be recorded.
That is why this class is about bacteriology of air. So we'll not only discuss about bacteria, but also spores of fungi, viruses, which can be there, which can spread from the environment. So first we'll see two videos about how to take samples from the air. Okay. So I hope that will be interesting. So let's see the videos.
And it's useful for evaluating the efficacy of cleaning disinfecting procedures. Surface sampling is performed using a general growth media, such as tryptic soil, supplemented with neutralizing agents such as linthicin and twine. Surface sampling devices must have a raised, convex surface. Each classified area of the aseptic processing environment must be sampled. The procedure per USP 797 is as follows. Label the sampling device with the sample location and sample date. Remove the cover from the sampling device. Using a rolling motion, firmly press the media onto the surface to be sampled. The media plate will leave a residue on the sampling site, which should be removed after sampling using a sterile 70% IPA. Cover each sampling device and lock the lid into place to ensure the lid stays secure. Store the media inverted during incubation to prevent condensation from dropping onto the agar. Samples must be incubated in a calibrated incubator at temperatures that will promote growth of bacteria and fungi. The incubator temperature must be monitored during incubation, either manually or by a continuous recording device, and the results must be reviewed and documented. Sterile swabs, whether with sterile water or sterile neutralizing agar, may be used for sampling irregular surfaces and difficult to reach locations, such as crevices, corners, and spaces between surfaces.
Okay. So as you saw in this particular video, what they were doing is they were sampling surfaces. Okay, so that is what we actually do in operation theaters, in labs, where the environment has to be sterile. So what they do is they will take swabs, they will put the swabs into inoculating media, and then once they inoculate onto media, they will incubate the media, and then they will see whether any organism is coming and whether any particular dangerous organisms also there, like Clostridium tetani or any such, which can be of very much clinical significance in patients. Okay. So another thing also we'll see about sampling of air. Those things that you saw in the video, we actually do in the OTs also. So they will sample surfaces and then they will particular media into particular media they will put them.
Two sampling techniques are used to search for particles or microorganisms in the air: particle count and aerobiocontamination measurement.
La surveillance de l'air dans les établissements de santé devient bénéficie par des contextes. Les objectifs de qualité d'air sont disponibles dans les normes NFEA ISO 14681, NFEA ISO 14698 et NFS 90351 ou les recommandations PST d'usage sur l'exploitation. Les établissements de santé peuvent reprendre ces objectifs pour définir des personnels et des valider en contexte particulier comme les épidémies et les ressources humaines. Les contrôles d'air aussi justifient en zone à environnement maîtrisé et s'effectuent tout le long de la vie de ces zones sous la forme soit de qualification opérationnelle effectuée lors de la mise en service de la zone à environnement maîtrisé ou de requalification annuelle. Elles doivent être réalisées par des entreprises extérieures, soit de contrôle de routine, de rapproche sujet qui permettent en gros de tracer une cartographie de suivi des points critiques participant à un processus de maîtrise environnementale dans le cadre de démarches techniques.
Technician complies with the controlled environment stressing and hand hygiene regulations. particle count sampling will use one particle count. Position the particle count on a flat surface at a comfortable working height, either with the probe facing the flow for a unidirectional flow, or with the probe pointing upwards for a non-unidirectional flow. Carry out the count in compliance with the pre-established sampling plan. If necessary, wait for the particle level to stabilize before starting the count. Measure at least 1 micrometer for the ISO 5 and 0.5 micrometer for the ISO 6, 7, and 8 classes. Keep still or move out of the area if the particle count has a delayed start function. Retrieve the data from the particle count. The technician complies with the dressing and hand hygiene regulations. For aerobiocontamination measurement sampling, we'll use one aerobio collector, one nozzle, one agar. Place the biocollector on a flat surface at a comfortable working height. Open the agar. Position it in its place facing upwards. Then screw the nozzle. Start the device and set the volume of air to be sampled. 1000 litres for M1 and M10 classes, and 200 or 500 litres for M100 zones. Keep still and move out of the area if the aerobio collector has a delayed start function. As soon as the sampling is done, close the agar case lid down. Hold this position so as to avoid that any condensation of the incubation period falls on the agar. For more details on each sampling, consult the CLASS Sud-West publication, Environmental Microbiological Monitoring in Healthcare Facilities.
Okay, so as you saw, we have to sample the air, we have to put a plate and then collect it. Okay, so we'll discuss about the general characteristics that are there in the environmental sampling, as well as what organisms can be there. Okay, so Aero Microbiology was a term which was used to denote airborne spores like fungi or other microorganisms during the 1930s. Then later this term was elaborated to include discussion of insect population, fungal spores, bacteria and viruses. And then there was research work of airborne materials of biological significance also, which was included in this term. So usually these are microorganisms and their spores which are invisible to the naked eye, which are suspended in air, and the study of aerosolization, aerial transmission, and biological materials. It also includes the study of diseases which are transmitted through respiratory routes, and it is important as it involves the formation of aerosols, especially in the era after COVID-19 has started. So their transmission and deposition in soil, so all these things are important. So there are different gases also which can affect the microorganisms that are suspended in air, that is hydrogen sulfide, sulfur dioxide, carbon monoxide, chlorine, hydrogen fluoride, ozone, etc. There can be bacteria, fungi, actinomycetes, algae, and different spores, pollen grains, different insects, and viruses which can be present in air. So different bioaerosols are there, as you might be knowing in this particular situation. All of you are aware about aerosols, droplet transmission, dry dust particles, and droplet nuclei. So different organisms can be sprayed by coughing, sneezing, etc. And these microorganisms can be carried through dust particles or droplet nuclei. So they will condense onto droplet nuclei and then they can be carried through air. So in the physical environment, different stressing factors are also there onto the microorganisms, which can affect whether they are present or not, like desiccation, humidity, temperature, radiation, And in indoor air, especially organisms like tubercle bacilli and all can be present. So in outdoor air, different organisms like algae, protozoa, yeasts, molds, etc. can be present, especially if you go into a room or a space where it has been closed for a very long time. So you can smell that particular moldy smell. So which is not always healthy. So that is why your parents will always say that you open the doors, windows, so that air transmission will be there. Okay. So bacterial species can be spore forming, non-spore forming, different bacteria can be present. So Micrococcus, Sarcina, gram-negative rods, gram-positive rods, or aerobic spore forming bacteria can be present in the air. And different airborne bacterial diseases also are present, like diphtheria, which was actually a very important disease in the pre-immunization era, where DPT vaccine and all were not available. So there was a formation of a fibrous pseudomembrane on the respiratory mucosa. There was effect to the myocardium and neural tissue damage, and there could be sore throat, low fever, cutaneous lesions, etc. So diphtheria was actually a very important disease before the DPT vaccine was administered. So tuberculosis, which can be caused by Mycobacterium tuberculosis or other non-tubercular mycobacteria, it can be initiated by inhalation, chronic cough, blood-tinged sputum, and night sweats, weight loss, etc. would be there. Meningitis due to Neisseria meningitidis, where there will be inflammation or infection of the meninges. Different symptoms like headache, neck stiffness, fever, confusions, photophobia would be there. So different airborne viral diseases are also there. Smallpox was once upon a time a very important disease. It has been eradicated thanks to vaccination, caused due to variola major and minor. And then it was localized in the small blood vessels of the skin and mouth, and there would be rash and fluid-filled blisters. So measles is a contagious disease. Even now it is present. There will be fever, cough, conjunctivitis, eruption of the buccal cavity or labial mucosa, and it is caused by a paramyxovirus and spread by droplets. So influenza, even now it is important. Even if COVID or any other disease is present, even then influenza is present. So it is an acute respiratory disease which is spread by orthomyxoviruses. So the different airborne fungal diseases are also there, like histoplasmosis, which is an infectious disease caused by Histoplasma capsulatum, also known as Darling's disease, and primarily affects the lungs. So cryptocorposis is a infectious disease caused by Cryptococcus neoformans, and people who are living with HIV/AIDS, it is a very important disease. It primarily affects the lungs, meninges, kidneys, bone, and the skin. And control of microorganisms in the air can be done by ultraviolet radiation, as you know, we always do in the hospitals. So by direct irradiation, like aseptic filling of the rooms for pharmaceutical preparations, or by indirect irradiation in occupied rooms or offices, wards, etc. So chemical agents, different agents are there, like you discussed in the chapter of sterilization, disinfection. So they are usually germicides and highly bactericidal. So they eliminate the microorganisms. So as you know, we do fumigation of wards, of OTs, of labs. So it should not, this chemical should not stain, discolor, or damage objects. And different agents are available which are used to make the environment free of microorganisms. So you might have seen in our lab, we have laminar flow systems where air passes through higher efficacy particulate air filters. So these are Hepa filters which are attached to the laminar airflow where we work. And if not, what will happen is we'll have the air which is contaminated by microorganisms, which will contaminate the media, and thus we'll not know that actually the media which we are inoculating is contaminated from the air or from the sample which we are inoculating. So we use them for different HEPA filters are used not only in these laminar airflow systems, but also in different rooms, especially biosafety level 2 or 3 rooms. We have HEPA filters where we have to work. So aerosol deposition depends on gravity settling. So naturally if the particle or the particulate matter is very heavy, naturally it will settle down due to gravity, surface impaction or rain deposition. So you might have seen that after rain the air appears very clear. Yes, so that is because the rain has cleared up the environment. So indoor environments like laboratory and all, there are different systems like filtration or HEPA filters or laminar airflows which are present, radiation, proper ventilation, biocide control or fumigation. You might remember with formaldehyde and potassium permanganate, we fumigate the rooms. So physical isolation. So these, as they discussed in the video, we have different norms which we have to follow. So according to those norms, we have to uh, clean the air or the environment in which we work. So we can sample by active sampling or by passive sampling. So environmental sampling is not exactly clinical microbiology, but it is supported by epidemiologic assessment, as we saw in the videos how they were collecting the samples. So there is a protocol for sampling and culturing analysis of the results. So suppose there is any outbreak, for example, now we are going through a pandemic for COVID. In such cases, it is very important that the environment is clean and safe enough for everybody. So suppose we are doing research in environmental infection control, or suppose we are monitoring a potentially hazardous situation. For example, people when patients were suffering with Ebola, the other cases. So we have to check whether the environment is safe or not. So then we have to periodically, regularly monitor the equipment like AC systems and other things, so that it is not a hazardous thing. So it is actually very difficult because there are no baselines or no acceptable ranges, but still we have to minimize the false negatives and false positives. So first we establish baseline data, for example, in the OTs. So usually we have to see that number of organisms are not there, and potentially dangerous organisms are not there. So that suppose there is any construction or repair of certain buildings, after that especially we have to check that whether that thing is clean or not, and microorganisms are not there. So because air is not sterile and does not support the growth of organisms, we have to compare indoor results to counts from the outdoor air, and then high volume sampling is most important. Like we saw, they have a nozzle in that particular instrument, and then all the air is sucked in and then impinged onto the agar medium. So impingement can be done on liquids or on solid surfaces, or simply we can put a plate. For example, we have to keep a blood agar plate for about 1 hour in the center of the room. That is known as a settle plate method. So whatever organisms are there, by gravity, they will settle down. Okay. So impingement on liquids is, the principle is air is drawn in through a small jet and directed against a liquid surface. So viable microorganisms, water aerosols, etc. And these organisms can be measured by collection on different media or surfaces like peptone water or nutrient broth or cryptosaline broth. So Legionella, etc. sampling, suppose we are staying in AC rooms and all. So it is very important for us to check. Then there is another method which is impaction on solid surfaces, in which air is drawn into the sampler and the particles are deposited on the dry surface like we saw in the video. So viable microorganisms, particles, all those things we can see. So the collection media are dry surfaces or coated surfaces or agar media, and bacteria, fungal agents. So high volumes can be sampled because air is drawn in through a nozzle. Then there is a sedimentation or a settle plate method, in which through gravity, the aerosols or the particles which are suspended in air, they settle through gravity. So nutrient agars in plates or slides, those are best suitable for qualitative sampling, but not used usually for fungal spores. So here you can see these are impact. This is an impactor sampler where air is drawn in through one nozzle. Then this is a glass impinger sampler, and this is a SEV impactor sampler like we saw in the video. So this is the nozzle. So this is like a SEV. So what happens is because of lack of standard sampling protocols, even though we have standards, it can be highly variable, and then we have to correlate between the different organisms that we isolate. So here you can see an auto sampler or a high volume vacuum pump is used, and a surface air sampler. So this is what usually nurses, the sisters who are in charge of infection control, they will go to different places, like they go to different wards, they will go to different OTs, and especially before and after fumigation, they will check. In OTs, it is very important. In labs, it is very important. So passively also we can check via the settle plate method. So simply one agar plate we have to keep in the center of the room, and then we have to keep it open for some time, and then later on we have to bring it to the lab, and then we have to incubate it, and then check what organisms we have got. So how to generally control airborne diseases is, first most important is the good ventilation of the room and avoiding overcrowding, especially in the post-COVID situation. Once COVID has started, we have remembered about social distancing, especially in closed spaces. We should avoid overcrowding. We have to isolate patients with serious respiratory infections. Those people with influenza, other important respiratory diseases, they have to be isolated. We have to wear masks and then disinfect air through HEPA filters or UV hoods, and then vaccinate. Okay. So this is how the after incubation you can see that they are agar plates. This is a nutrient agar plate. So different organisms like fungal spores also can be got from the environmental sample. So these are actinomycetes which can be also found on agar plates, which can be suspended in air and later on can be found. So these are actinomycetes, gram-positive rods, you can see. So yeasts also we can get. So thank you all for patient listening. One short video now we'll see. After that, we can finish the class. This is how they are fixing the agar plate. This is by impingement onto a solid surface. So this will spin, and then it will draw the particles, like you can see here. The particles are being drawn and impinged onto the agar medium. So because it is rotating, so that is why. So here you can see this is the safe like nozzle. So once it will rotate, it will help in impingement of the particles onto the solid agar surface. So using a tripod, whatever height you need also, you can sample the environment. So thank you all for joining us today. I hope you are studying properly. Go through all the systematic bacteriology chapters, mycology, then virology, all the chapters. And if you have any doubts, you can always ask. Thank you all. Have a good day.